for such a simple molecule, it had very widespread effects in the central nervous system, affecting brain development, metabolism, and immune function. And then, we sort of put two and two together, and we said, hey, these bacteria in the gut, when children eat carbs, ferment the carbs and make propionic acid. And in addition, some of the foods that these children craved, in addition to being high carb, did in fact contain propionic acid as a food preservative.
Propionic acid is labeled on food packaging as E280. It's a commonly used preservative in many bread, cake, and cheese products made and sold throughout Europe, Australia, South Korea, and North America.
So we thought to take propionic acid and give it in small amounts to rodents and see if the behaviors or brain changes looked anything like autism and whether or not these behavioral and brain changes were transient.
Dr. McFabe's control subjects exhibit typical behavior. Rats are highly social animals. They greet by sniffing nose to tail and interact as they explore their surroundings. But when a propionic acid solution is introduced, this all changes.
What we noticed remarkably within 2 minutes of putting this propionic acid in, this animal immediately starts exhibiting some very peculiar behaviors. Obviously, he's hyperactive and moving very quickly, and then repetitive behaviors. Here you see an abnormal posture of walking with a hunchback. And it's an unappreciated part of autism in that patients often have toe walking, abnormal posturing, arched back. So now we'll place another animal that's been given propionic acid. And a peculiar thing happens. They show none of the normal social greetings of two rodents seeing each other.
A video tracking system records and compares the obsessive and antisocial PPA-treated rats with the normal rats. Other experiments showed responses such as rigidity and object fixation, and the intriguing fact that the effects of PPA wear off.
So now we're about a half an hour after we've exposed these two animals to propionic acids, and their behavior afterwards reverts back to normal, and it's a fat, so it's burned through metabolism. The animals become more social. Usually after their escapades, they tend to just go back to sleep. So again, we have a reversibility of hyperactivity and a reversibility of social behavior.
The autistic-like behavior of PPA-treated rats was compelling. But Dr. McFabe and his team wanted to know exactly what PPA was doing to the brain.
So do you have the lipids on the PPA rats?
Yes, here it is. This is what the final product looked like. When I finish this analysis, we'll be able to quantify whether or not there's any alteration in any of these components.
Brain fat composition is intertwined with behavior. So Dr. McFabe is working with lipids expert Dr. Ray Thomas, who's been examining PPA's effect on lipids, the fats that fuel brain cells.
What we've found is that when we give propionic acid, which is a metabolite from enteric bacteria, it has the propensity or the potential to alter brain fats. They alter the omega-3s, the omega-6, the saturate level. And these alterations that we've found in the brain is closely related or similar to what has been reported in the blood of autistic patients. What we've shown is that with propionic acid, you get a fast change.
Remember, families are saying, my child gets worse with something associated with gut or diet. And sometimes when I make manipulations with diet or gut, my child gets better. We wanted a model that gets worse and better. And what Ray found were these amazingly specific changes in very key lipids that have to do with brain development, brain functioning, and brain energy metabolism. Imagine these gut bacteria produce the propionic acid as a metabolite. It'll fly through all bloodstreams to multiple cells, like all the other fats. But then when it comes to the nervous system, it can get in because it's a small molecule. But there are actually very specific compounds called transporters that actually grab the propionic acid and kind of suck it into the nervous system.
Once in the brain, PPA changes brain fat composition to be similar to that of autistic patients. The brain is 60% fat, and cell function and our behavior are directly related to how those fats are composed. And there's more. PPA could even be switching the genes associated with autism on and off.
Imagine this little molecule from your gut going directly into the nucleus where all your DNA is and acting as a switch to turn on and off important genes having to do with brain development, learning, and memory and metabolism. It's very intriguing that such a simple molecule can do such specific things in a brain metabolism and function that are very consistent with what's found in autism.
So far, there are no studies showing that PPA or E280 isn't safe for adults when used as a preservative. But Dr. McFabe's work with animals shows that gut bug metabolites like PPA could be triggering autism in susceptible children.
Normal or near normal people exposed to high levels of alcohol are not behaving as their normal selves. And by analogy, I think normal or near normal children exposed to high levels of propionic acid, gut bug metabolites, are not themselves. By removing the propionic acid, by either getting rid of the bug that makes it, not feeding the bugs that are there, healing the gut, or using compounds that help break it down, that will be the approach of treating this disease.
Yeah?
Oh, pop, pop.
Oh no.
It's painful, to be honest. It's painful to watch it because we were so close. I really think that we were so close a decade ago to fixing this, but no one would listen.
Many parents have taken up the gut bacteria theory and are using it to devise treatments.
Andrew is now 19 years old. It's hard to believe that it's been that many years. I do think that it's a war in Andrew's intestinal tract. There is a colony of, I believe, neurotoxin-producing bacteria that established itself and set up camp and set it up good and strong and at an early age. And basically what you become colonized with as a child, those are the bacteria you die with.
Keeping Andy on powerful antibiotics indefinitely wasn't an option. Long-term use would have dangerous side effects, so Ellen turned to other therapies. She has found that by giving Andy massive amounts of probiotics, supplements containing benign bacteria, his symptoms are controlled. So even if Andy's Clostridia can't be eradicated, they can be beaten back with waves of competing bacteria. Studies have confirmed that probiotics have helped other children as well.
He functions very well, and that's a relative term. When I compare us to our baseline, I'm very happy with what he can do. Andrew is very self-sufficient. He can cook his own breakfast, pack his own lunch. Academically, he struggles beyond, a third or fourth grade level. He still has very impaired social skills. He is, though, delightful and a joy to be around the house. Andrew, pretty soon you have to go to your track meet.
Hey,
And what are you going to do at your track meet?
run fast.
Run fast.
Besides hopeful signs that altering gut bacteria may control autism, new research in Canada is looking at the possibility of medically replacing a whole internal ecosystem with a healthier one.
This to me is like a golden opportunity to really look and see what might be going on in this situation, where I can sort of see that there's some problems with the gut and problems with development.
Dr. McFabe and his colleague, Dr. Emma Allen Vercoe from the University of Guelph, have begun a unique collaboration with the Somali community in Ontario. Joining forces with parents, they've started a study that will analyze fecal samples from Somali Canadian autistic kids. Dr. Allen Vercoe hopes it will reveal what's amiss with their bacterial profile.
And also this entire change that the Somali population has when they come from Somalia to Canada, to a completely different country, a different culture, different foods in particular, and hearing the changes that might be happening in the diet that might therefore have some kind of knock-on effects with the bacteria that live in the gut. The old adage, we are what we eat, I think that's very important. In Somalia, they eat a lot of fermented foods, and that's a very staple part of their diets. They wouldn't necessarily do that in Canada.
It's the way we lived, you know, the food we used to eat. No corn, no soy, no genetically modified things, no nothing. Everything is as natural as they can be. No pesticides, no hormones, nothing.
And maybe they have a microbe that's missing now from their diet because they're not getting it from fermented food. And maybe that microbe might have some direct interaction with other microbes in their gut. Now, if that's a missing piece, it's that no longer there. Their gut microbiota might actually not be functioning correctly anymore.
Dr. Alan Vercoe is working on the idea that the ecology of gut bacteria could be medically manipulated.
Over here, we have the RoboGut, which is a very sort of coined phrase for our artificial gut system that we have here. Now, here we're able to actually recapitulate the environment of the human gut, which enables us to culture some of the organisms just by growing them with their friends, essentially, as part of an ecosystem.
Dr. Alan Vercoe approaches her work as an ecologist, using the RoboGuts to create whole microbial communities, then manipulating them to see what happens to the stability of the ecosystem.
It's an ecosystem that is incredibly diverse, in fact, probably one of the most diverse ecosystems on the face of this planet. And I liken it to a rainforest. Everyone knows what happens when you start stripping species out of an ecosystem like a rainforest. You end up with a barren sort of landscape, and really the ecosystem just kind of crashes. And this is what we think is the kind of the basis for many diseases now.
The concept of treating gut microbes as an ecosystem leads to hope that someday a whole flawed set of microbes could be replaced.
The idea would be that we would get rid of the bad ecosystem with antibiotics, with purging of some kind, and then replace it immediately with a healthy ecosystem that would allow that to establish and to take on the job of a healthy ecosystem.
So autism can be a shortage of some microbes and not a presence of some microbes. And that's difficult to investigate, but you have to do it.
In Bergen, Norway, practical work in devising therapies for gut-related autism is becoming a science. And it can be seen in Tor Midtvedt's own family. Dr. Midtvedt's nephew, Lars Ørsland, and his wife, Britt, have 2 sons. The younger one, Eric, is autistic.
How old was he when you first made the diagnosis?
He was 6 years old when he got the diagnosis.
Okay.
But since he was 2.5 or something like that, he started to slip away. We didn't feel that we really had contact with him. But that was after he had some stomach illness when he was about 2.5.
Today there is another visitor besides Dr. Midtvedt, Hanna Bjorg, a biochemist. Bjorg gave up a successful career in banking to study how to treat autism biomedically.
Well, the history of why I'm working with autism at all is that I have 3 children that also have needed a lot of help. And we went through 17 doctors here to try and get help for the children. And none of them could help the children.
So, Hanna, what do you think about this test?
Bjorg traveled to America to find answers and was able to improve her children with what she learned. She decided to bring that knowledge home to Norway. Bjorg co-founded a clinic in Oslo, one of the first in Europe. Its mandate is to treat autism by treating the body. It's an idea that is still relatively new.
The official statement is that in Norway is that autism is not treatable. That's the official statement in Norway. It's something new for the medical community, but probably not for the parents who's been working with kids for a long time.
With every new case at her clinic,